Thank you for attending today. Today, I would like to introduce you to Dr. Joseph Buxbaum and his team from the Seaver Autism Center for Research and Treatment at the Icahn School of Medicine and Mount Sinai in New York City. Dr. Buxbaum is the director of the Seaver Center. The center conducts research studies and provides care to children and adults with autism spectrum disorders. And they work to understand the biological causes of ASD and to develop treatments, as well as to provide education and training opportunities. Dr. Buxbaum is considered one of the world's top leading authorities in genetic science and is a professor of psychiatry, genetics and genomic sciences, and neuroscience. The center already studies several rare genetic neurodevelopmental syndromes linked to ASD, such as ADNP, DDX3X, FOXP1, and SHANK3. And on the call, we also have Dr. Shaper from the Seaver Center. And we've gathered here today to hear more about their upcoming research that will include SYNGAP1 related disorders, and to share your stories about SYNGAP1 with the team at the Seaver Center, and to answer any questions you may have about the center and its upcoming research. And just a quick note on housekeeping. I know we may have many families joined across the US and internationally. However, because the center is based in New York City, we would like to prioritize the Q&A airtime for the families based in the New York area or who can travel to the New York area. And with that, I will hand it over to Dr. Buxbaum and his team. Thank you.
Thank you, Jess. Happy to be here. I have slides because I need slides as a crutch. But we'll use the slides to kind of introduce these three studies that we are planning on SYNGAP1. And what I will do is I will share my slides. And what are we seeing? Are we seeing the proper view? Not yet, I gather.
Yeah, I think you just have to switch to slide view.
How's that?
Perfect.
Okay, good. Yeah, so we'll talk about three studies, one that is kind of drug development and two that are involve families and patients. I'll talk about the first one because that's all I really know. But very briefly, the Seaver Center is kind of it's an integrated research, clinical research, translational research, and clinical center. It has a lot of areas of expertise and faculty that all work together. And the idea is in the rare disorder space to kind of work with family foundations to become experts on the disorder, thinking about clinical care, but also endpoints for clinical trials. Take advantage of model systems like in vitro model systems. Nan Yang is on, she's an expert at this. Animal models that may have a mutation in the gene. I know there's work in that space. And ultimately come around to do clinical trials. And by having experts who agree to all work together on a single gene from these different perspectives, we're able to move kind of across that arc of translation. I was just asked to put this in. I just got this to put this in. We have an associates board that tends to include many of the people on it are from rare disorder communities, and they kind of help give us direction specifically for that rare disorder. We are just today had a big meeting about re-upping our associates board. If you want to hear more, just reach out. There's a commitment of time and energy and other things like that, but it also gives an opportunity to have, to get feedback from profound autism, for rare genetic disorders, and so on. So, Jess mentioned some of the genes, ADNP, FOXP1, DDX3X. And like I said, what we try and do is we try and pair clinical studies, patient-based studies with preclinical studies. And so, families will come in to get these multi-day assessments, natural history studies. That helps us to understand, clinical endpoints, clinical need for intervention, how things change over time, right? Predict changes and become actually experts on the disorder. And we often write practice parameters for non-specialists and so on and so forth. And in parallel, we try and make models in Petri dishes or in animals with a genetic mutation, understand about the biology, and maybe those studies will suggest drugs for repositioning or repurposing that might be tried, safe drugs in clinical trials. But ultimately, also, we want to come up with new drugs that are specifically targeted for the disorder we're talking about, and then come up with novel chemical entities. And by having experts that work across all of these domains, we're able to kind of hopefully move that quickly. We choose our genes based on, very compelling evidence. We're actually one of the founding lead sites for the Autism Sequencing Consortium, which is an NIH-funded program to discover genes that are associated with autism and profound autism. And we've been doing this for quite a while. And over the course of time, every couple of years, we have kind of flagship papers that talk about some of the top genes and what we know about them. It won't surprise you that Syngap, one of the top genes over and over again. It's a gene we're very interested in for various reasons, not only partly because it's a top gene, but partly also because it's a synaptic gene. And we have, you've heard us, we work on SHANK3, which is another synaptic gene, and there's a lot to be said for looking at those genes in parallel. It won't surprise you that there are all these many genes, about the discovery in autism and profound autism and intellectual disability. As I said, SYNGAP is one of the top ones. And the reality, what we're now realizing is even though there are many, many genes, we now have about 250, the top 15 or 20 account for about half of all the mutations we're going to see clinically. And so there really is more and more impetus, and we're pushing this, and we're actually, going to funders about this, shouldn't we look very exhaustively at these top genes because there is such a critical need in the community. And if we're successful and we can figure out ways to develop therapeutics quickly, we can help everybody, not, all the way down the list, so to speak. And again, you already know this, but most people don't. The mutations in these genes are very, very penetrant. They are the main cause of the phenotype in the individual, right? And so this is from a review, but basically there's SYNGAP1 right there. So it's right about up here. The relative risk for autism in this case is over 50, but it's really for what we call, what you all know as kind of profound autism, or the autism part is really not the key. It's the cognitive, communicative problems and the daily living skills problems. And so when you have a mutation that increases risk for a phenotype, by 50 or 100-fold, that's explaining most of the phenotype. So if you could take that mutation away, right, you should actually do amazingly, have incredible benefits for the individual, right? And so each of these genes, as we find them, they really are basically, by kind of legal definition of the FDA, they're actually orphan genetic disorders, which means that they have special, opportunities for fast tracking within the FDA program or the EMA program. They have opportunities for actually getting money back, special vouchers that will help support rare disorders research. And so, it becomes, an opportunity really. I know it's a sad way to talk about any of these disorders, but those tools and those resources created for these rare disorders were meant to address the fact that they are rare and often not at the very forefront of what a big pharma company is going to be thinking about, right? From the point of view of drug development, when you have a genetic disorder, you can actually make human cells that have that mutation or animals that have that mutation, right? So we do all, we study and identify the genes. We try and make neuronal models. We try mouse models. We see patients. We understand the phenotype, but then we also want to bring drugs to the patients to improve quality of life. So the three studies we'll talk about. The first one is small molecule targeting, which is a drug development strategy, which I'll talk about. And then Paige will take the next two, which have to do with patient-based studies, a natural history study, which is through the RDCRN, and a sensory processing study, which is her expertise and has been for many years. So the small molecule targeting of SYNGAP1 for drug development, right? So, this is the same figure I just showed you, but now we're putting in a therapeutic approach, right? So if you have a gene, SYNGAP1 in this case, and the gene is, you want to, the gene is missing, you want to fix it or replace it, you're thinking about a gene-targeted therapy. I avoid the word gene therapy because people think about that in a very specific context of viral vectors. More broadly, we're targeting the gene that's mutated to try and improve the situation, right? And then we can use those same Petri dish-based models or animal models to test our gene therapy, our possible therapy, and ultimately come back to improved care. If you need to interrupt because I'm confusing you, please don't hesitate. I don't know how you do that. I'm sure you have to put something in the chat, which I'm not watching, but somebody tell me if there's a question.
Yeah, I'm watching the chat. I don't see any Q&A questions so far, but yes, anyone, if as Dr. Buxbaum or any of his colleagues are going through these slides and you have questions, feel free to put them into the Q&A box in Zoom and we'll get to those. We won't miss any of those. Thank you.
So this is a slide I showed you before of how we work, and I realized I say across laboratories. It is true. All the laboratories at the Seaver Center work across modalities, but we also do a lot of work with outside collaborators, and that's really important for this study I'm talking about because we are funded and we've worked very closely together with Jessica Charles Disney and Matt Disney for this drug development program that we think is has the possibility of getting to drugs and genetic disorders much more rapidly and much more efficiently and probably better than other methodologies. I'll explain the approach in painful detail. Nan Yang is on the call as well. She's our human neuronal modeling person. She's also formally part of this grant as well. But the idea is if we can find small molecules, basically drugs, oral medications, right, that can bind to the gene of interest, this would be SYNGAP1, and a particular region of the gene that regulates its expression, changes the amount of product the gene makes, then we can take a gene like SYNGAP1 where there's one mutant copy and one intact copy, and we can ask the intact copy to make more of itself. So instead of having half as much SYNGAP1, this small molecule, this drug, is going to tell the gene to make more, and so it compensates for the loss of one of the copies. And the prediction is that because that mutation is accounting for the phenotype, that it will rescue some of the neuronal function, the behavioral function, and ultimately have a positive impact on quality of life. So that's the strategy, right? And one of the reasons we're really obviously a small molecule, non-invasive, take it in your mouth, right? If it works. The screening is much simpler. We don't have to inject something in the spinal cord or in the brain like a gene therapy would. If the small molecule we're very good as a community of figuring out how to make small molecules get in the brain, how to make them safe. And so there's a way that this can move pretty quickly. But the other thing about small molecules is this big challenge in gene therapy, which is that we're pretty sure that you don't want too much of any of these genes, that too little causes a problem and too much causes a problem. And so we are very worried about how do you kind of gradually get to the right dose and not overshoot it. And if you're doing things that are very, interventional, like injecting to the spinal cord, and each time you inject, it's a little bit different. In fact, we all now know that it's quite a bit different. Each injection, how effective it is, changes from injection to injection. If you put in a viral vector that's irreversible, whatever you put in, you put in. You can't add more because you develop antibodies, and you can't take it away, right? So the small molecule gives us the opportunity of maybe, coming slowly rising up to a most maximally effective dose and not surpassing it, and just taking a pill to do that. And this is called the Goldilocks effect. It's true for many genes. You don't want too little, you get a syndrome, but you also don't want too much, because that also will probably produce a similar phenotype. So the idea is, if you can take a reporter, and this is the methodology developed by the Disney lab, if you can this is an enzyme. This is luciferase. This is from the firefly. So it gives off light. It's an enzyme that gives off light just like a firefly does. You take part of your gene of interest, okay, and you then can use that construct to screen many, many compounds, 5 to 20,000 drug-like compounds, and ask which of these drugs um, make more light, less light, or no change. So this weird gene construct has the enzyme from the firefly that will give off light. And if a drug forces that gene to make more of itself, you'll get more light from that in that cell. If it somehow makes it make less of itself, you get less. And if it has no effect, you'll get no change. So basically what you can do now is you can actually screen 5 or 10 or 20,000 compounds quickly because you're basically looking for more gene expression simply by measuring light output in that subset of cells that are getting exposed to that drug. And the drug-like library, which is developed by the Disney Lab, really has a couple of properties. One is it likes to do what we need it to do. It's similar. It has properties that are drug-like, and also properties that that can actually say it'll get into the brain quickly. And I'll give you an example of one of the genes right next to SYNGAP1, ADMP, this one over here, which we've been studying for a couple of years as a collaborative study with Nan and the Disney Lab. And we did all of those things. We did all those things I just showed you. And here now, this is the protein, not the gene. But these are two drugs. This is one going from low to high. This is another, P1A9, going from low to high. And what you can see is that the protein that was missing in these individuals, we can make the cells make more and more of it as we increase the drug. So ADMP syndrome, just like SYNGAP1, there's a mutation in one copy and the other copy is intact. And what we're trying to do here is we're trying to say, okay, we've lost some ADMP. Can we make more from the intact copy with a drug? And here we have two compounds. These are not these are drug-like or they're not even drug. These are drug fragments, if you will. And you can see that the darkness of this band is telling us how much ADMP is in that cell. And what you can see is we can push it up with this drug and we can push it up with this drug, right? So then what we want to do is exactly that. So we've definitely done this with ADMP. And now we're checking this. We have ADMP neurons in culture and an ADMP mouse. And so now we can actually measure, do those drugs actually do something positive for nerve function and animal function. And so obviously we want to do exactly the same thing with SYNGAP1. And the first step is what I've shown you before, which is we have to make this luciferase construct with part of the SYNGAP1 gene and then test it and make sure that and see if it can be used in this kind of screen. So the first question is, does SYNGAP1 have these kinds of structures in the gene that might be amenable to target? And the Disney Lab has already looked at that for us. And basically, we're targeting RNA, and RNA has a lot of structure. It forms these, it adheres to itself in very specific ways that create these structures that then other things like small molecules, but also proteins can bind to. So the first question is, does SYNGAP1 have some of these stable functional structures? And the answer is it has quite a few, and it has them both in the 5' or the beginning of the gene and in the 3', which is green here of the gene, where most gene regulation happens. So there's plenty of opportunities in SYNGAP1 to target some of these structures that exist both at the beginning and the end of the gene and see if we can't regulate expression. Allison, is that a question?
Hi, yes. Hi. I have a question. Are you actually able to target the intact gene copy, or would you be targeting both copies? And would this be relevant for missense mutations as well? Since missense mutations have two technically working copies. One of them is just obviously not working properly.
Yeah. I would, many missense are actually loss of function, right? You're right. They're not working properly, but their missenses can be gain of function, dominant negative, or loss of function. And everything we know about SYNGAP1, here's SYNGAP1. First of all, most of the mutations are red. They're protein truncating variants, right? We have data from, this is from thousands of people. We now have data from 60,000 individuals. A certain number of people actually have a deletion in the gene, that's purple. That tells us that the most common mechanism is a loss of function. In other words, one copy is not working. And then you're suggesting that maybe the missense might be kind of working. I would argue that we don't think so right now. Not to say that we're right or wrong, but we don't think so, right? When the missense is doing some of these genes, you'll see most of the mutations are missense, right? That's these different colors here, right? So P10, most of it is missense. SLC6, most, and these are actually dominant negative or other mechanisms, right? So missense, more often than not, is functionally not working rather than working wrong or working weirdly. This is something that we worry about all the time. I'm not saying your question is not excellent. It is. For every disorder we study, we spend a lot of time in that space to try and understand exactly, how the missense are working. Right now, we would target both alleles, and we don't expect nothing deleterious from the missense allele, but one can also make specific approaches. Great question.
Thank you.
Imperfect answer, right?
Thank you.
And so the structures are there that could be targeted with the small molecules. So Jess has shown that. And then the next step is to actually create this construct. So basically take this, take this enzyme from fireflies. I love fireflies. That's why I mention them all the time. Add part of the SYNGAP1 regulatory region, and then see if you get expression of light relative luciferase units, and then is it dose dependent, and does it have all the properties you want to be able before you do a screen of 5,000 compounds? And these two figures here are basically confirmation that the Disney Lab has created a construct that if you increase the number of cells, you increase the amount of this construct, you get more and more light in the way you want to see it, and at a level that now one can screen 5, 10, or 20,000 compounds relatively. I'm not saying it's easy, but relatively easy to the point where we can do a screen and come up with some hits, and then go through this process of pruning down the hits, making sure that they're specific, understanding how they work, making them better and better through a method called structure-activity relationship, make them more drug-like, test them in the neurons in culture, animals, and so on. So that is where we are with this study. We've just really convinced ourselves that there's something very targetable based on sequence and conservation, and that in fact we can develop a reporter construct that can be used for screening the libraries. The libraries already exist. Obviously, we've screened them before. The Disney Lab is in the process of massively improving the libraries to have a much greater chemical diversity. And, we'd like to work with the Family Foundation to actually launch a screen of the new library for SYNGAP1. And this is just the 3' this is the blue part of the gene, the green part of the gene, but in principle, we could also run a screen with the blue part of the gene, where the colors are what you see here if you're not color. So I will stop there. So this is, the first project around drug discovery and development. And I will turn it over to Paige. But first I will unshare. If I could just stop there. I found it. I found the stop share.
I will go ahead and share my slides. Thank you for having us. So now I'll summarize 2 studies, both we're actively recruiting for. One, we're bringing participants in. Another, we're building our list and we'll be ready to bring in participants. We're estimating at some point this summer. So starting with this sensory processing study, I'm just going to put the flyer up here. But basically what we're doing, and you'll understand the why in a minute, because I'll get into some of the details from a research perspective, but we're really trying to identify biomarkers of various genetic syndromes that we can then apply to clinical trials. So we've now done this in Phelan-McDermid syndrome, in EDMP syndrome, and have published in both of those on the biomarkers that we've identified. And so we're bringing in families to our center. It's about a 1-hour visit. We're doing something called a visual evoked potential, which you'll get to see what that is, and a behavioral sensory assessment, which is really fun for many of our participants. And so we're trying to understand both sensory reactivity, sensory processing, underlying neurobiology, and also objective markers that we can use to characterize a disorder. So starting with visual evoked potentials, VEP is extracted from ongoing EEG through signal averaging. So it's a type of EEG, but what we also do high density EEG at our center, where we put on several hundred electrodes. In this case, it's just 3 electrodes. It's called single channel recording. We're just recording over the visual cortex. And the reason that we're so interested in visual evoked potentials is less about the visual system and more about the relationship between excitatory and inhibitory postsynaptic potential. So VEPs are very well characterized, and animal models, for example, in the case of Phelan-McDermid syndrome, have been able to look at glutamatergic and GABAergic differences that we can also see in visual potentials. And we have specific stimulus conditions, so images that we show on the screen that probe specific mechanisms in the brain. And we have been working in this domain for a while and developed short duration conditions. So we know that our populations can be difficult to test. We know the value of getting objective data from difficult to test and profoundly affected populations. And so we have made various stimuli to make sure that we can get objective data from even the most profoundly affected individuals. And here's just a quick time-lapse video to get a sense of it. So we measure the head, we put 3 electrodes on, they watch a pattern on the screen, and this pattern will produce a brainwave that we can then look at and analyze. So some of the advantages of VEPs, they're non-invasive, data collection is rapid, we're able to get this on individuals of all levels of ability, we use only passive viewing tasks, there's no prerequisite language or cognitive skills necessary. It's inexpensive in that once you have the system, there aren't costs associated with continuing to use it in the way that, for example, MRI studies require. We can do repeated testing. It's no more harmful than watching the television screen. It's used on babies within the first few weeks of life for various issues, so it's very safe as well. So then when we think about applying VEPs to clinical trials, one, we're able to monitor treatment at the individual level. We can see what does someone's waveform look like before they've been given a specific treatment and how might that change. We can use VEPs to personalize treatment approaches, and this is sort of a bigger long-term goal, that can we look at dose response? So is there a certain dose where we're seeing change? We can determine optimal responders, so we don't necessarily expect that every single individual will respond to every treatment, but if we can figure out who are those people where we identified improvement on VEP, and what are those associations with clinical outcomes or specific genetic mutations. So for pretty much any healthy individual, this is what what we call a transient VEP will look like. And when we say the waveform is well characterized, there are certain points of the waveform that we really understand. So we analyze VEPs in 2 different ways. We analyze them in the time domain. So we're looking at the amplitude or how large the response is. We look at the latency. So at what time point does an individual hit these various, what we call peaks and troughs, these points in the waveform. And then we also look within the frequency domain. And these have been studied for many, many decades. So we know, for example, that when we take this measurement of the amplitude from the first positive peak at about 60 milliseconds to this initial negative peak around 75 milliseconds, that reflects glutamatergic or excitatory activity. And we know that when we take this measurement from this negative trough to this positive peak here, this is what we call P100 because it happens around 100 milliseconds, that reflects inhibitory activity. And so what we've been able to do when we talk about biomarkers and applying them to genetic syndromes is we're able to identify specific VP differences within a given syndrome. And I'll show you an example of that using Phelan-McDermid syndrome. So if you look on the top right, you have this control reference. You'll see two lines because it's showing the stability of the same person's VP at two different time points. And this is a control. And you can see clear differences in the waveform. So in these larger deletions, we see a complete absence of that negative peak. And in the point mutations, these scales are different. So I don't want to throw you off, but I wanted you to be able to see the waveform. In the last one, the responses are smaller than we would expect compared to a control, but you do see that sort of typical waveform. And this is a pattern that we see repeatedly over and over again in every single participant we've seen with Phelan-McDermid syndrome, and we've published on this as well. We recently also published on specific abnormalities in ADMP syndrome. And this is an example of how we've applied VEPs in clinical trials. So here's an example of one individual. So when we talk about how we can look both at the individual and the group level, you can see this individual's VEP response at baseline and how it changed post-treatment after 12 weeks of treatment with IGF-1. And then when we looked at the group level, we were able to look at the frequency domain. So we look at different frequency bands as another way to analyze data. And what we saw was that of these 6 participants who did VEPs in this trial, we saw improvement in 5 of the 6. And so when we think about responders, this is what we're talking about. We had 6 kids, or 5 of the 6 kids who appeared to respond to this treatment. And we could then look into, well, what were the factors that maybe were different in that one who didn't respond? So that's sort of where our VEP work stands. And that was something that I started working on many years ago. So starting in 2009, I got really involved in VEP work. And then in 2013, when the DSM-5 came out and sensory reactivity was first included as a core feature of autism, my colleague and I, Teresa Tavasoli, who was at Seaver at the time and is now in the UK, we developed a sensory assessment called the Sensory Assessment for Neurodevelopmental Disorders, also referred to as the SAND. This measure has been commercially available for a couple of years now. It's used pretty widely within the context of clinical trials, including gene therapy trials in various neurodevelopmental conditions. And the SAND is a behavioral assessment that allows us to quantify sensory differences. And so when I think about, okay, what's the link here? Well, we're oftentimes talking about how can we connect biomarkers to clinical symptoms, but really many of our biomarkers are looking at sensory processing. And so where we would expect to see sort of the most relationship would be with measures of sensory reactivity, rather than these sort of higher order social communication measures. And so we developed the SAND, which is a clinician administered observation with the child, as well as a caregiver interview that corresponds with that observation with the child. So anything that I'm coding as the clinician is then asked to the parent in a question and in the form of a question. So we're capturing the same behaviors. And the way I think about it is any sensory behavior that we see in a 10-minute observation likely happens pretty frequently throughout a child's everyday life, but there's many behaviors that we wouldn't see or that our stimulus don't probe for. So especially kids who might show certain hyperreactivities to specific sounds or textures that we might not see in this context. So we operationalize the DSM-5. We measure sensory hyperreactivity, hyporeactivity, and seeking behavior. So an over-responsiveness and under-responsiveness and seeking across visual, tactile, and auditory domains. These were domains that are well understood and that we could capture in individuals of all levels of ability. This was something that was extremely important to us when we were developing this measure, that it was a measure appropriate for everyone. Similar to the VEPs, no prerequisite language or cognitive abilities needed. Administration is straightforward, standardized. We have norms, and it's a continuous variable across ages. And we've published both on the SAND, but also have published on the use of the SAND and in clinical trials where we've seen improvement in sensory reactivity you'll see on the right, the Phelan-McDermid syndrome example I gave, where we saw the change in VEPs. We also showed that on the SAN, there was significant improvement in sensory reactivity, which was related to sensory or driven by hyporeactivity. And when we think about phenotypes of specific syndromes, Phelan-McDermid syndrome is characterized by very high levels of hyporeactivity, so a significant under-responsiveness, whereas we've published in ADMP syndrome where we see very high levels of sensory C gain. And so that's something that we also look at, sort of different sensory reactivity phenotypes within different syndromes. So for those interested in the sensory study, we're doing a VEP and the SAND. So that's the sort of 1 hour visit. There's $100 compensation for participation, and we are actively recruiting and bringing families in. So at the end, I will put up the contact information again. And I haven't checked if there are any questions, but we'll have time at the end. So the last study we'll talk about is the Rare Disease Clinical Research Network. Dr. Culbertson couldn't join us today, but this is the third cycle. So each funding cycle through the NIH is 5 years. We started 10 years ago, as the lead site for Phelan-McDermid syndrome, and are very excited that SYNGAP1 was added to the RDCRN for years 10 through 15, and we are going to be a site that is recruiting for both Phelan-McDermid syndrome and SYNGAP1. I'm just going to summarize briefly what the aims are, and sort of how this study has progressed over time. So the initial goals were really to characterize, in our case, Phelan-McDermid syndrome. There are several other conditions that were also involved, to identify biomarkers and genetic factors. We enrolled many participants. We collected some neuroimaging, biosample collection. A number of pilot studies came out of it. You'll see here just to get a sense of how many publications came out of these earlier cycles. Then in the second 5 years, we started looking across the lifespan. So we extended the age range into adulthood and to younger children. We had a larger focus on electrophysiology and continued with the clinical assessments. And this information has now been applied and is being used in a number of gene therapy trials as well. So it's really the natural history data collected is so rich in this from the RDCRN. And so now for this third cycle, we are going to comprehensively characterize PMS and SYNGAP. There's this focus on sleep and sensory reactivity, which are new. We will continue looking at EEG and the relationship with clinical symptoms, and we'll also look at neuropsychiatric symptom profiles and regression. And so this will involve sort of very comprehensive visit.
And everything.
With the goal of really developing this natural history database and information that can then be applied for use in clinical trials. The measures are not fully vetted for all of the different disorders, but just to get a sense of it, we'll be looking at the cognitive domain, adaptive behavior, communication, motor skills, sleep, as I mentioned. So we're really going to get a comprehensive look at each child. They'll get to come back a year later for a follow-up visit, and we'll get written reports as well with the results of all of the assessments that they do. And this just goes through some more details about some of the measures that are included. And so that summarizes both of those studies. If you're interested in the VEP study, where we can bring you in now, and we would love to bring you in for all the studies, so you can participate in any amount that you're interested in. I have our coordinator Stella's information here. If you're interested in getting on the waitlist for the RDCRN study, where there is funding that supports travel, there's also sites around the country. So based on where you are, our site may or may not be the most convenient, but certainly reach out and we'd love to get you into our network. And if you do have any general inquiries about anything related to research, clinical care, the Associates Board, I have my information up here as well.
The photo is, I'm just still looking for it. Never mind, I found it. Never mind. Okay, bye.
There you go. We bombarded you with a lot of stuff, but the good news for our perspective is that, we have our opportunity to bring in families to do sensory studies, which we think are going to be very important for understanding phenotype, but also for biomarkers. And the reason that study is funded in the first place is because we're not the only ones that think so. This is funded by biotech, actually, that are really excited about these approaches to measure as a means of quickly measuring improvement or positive directions to kind of reinforce a clinical trial. And obviously, sensory activity is an important clinical phenotype as well. And then the RDCRN natural history. I mean, that is, over the course, it's 3 years of ongoing studies per person. And so you get a lot of information about how the disorders in the natural study progress over time. And then, we think that the small molecule approach that, will add to the armamentarium of treatment modalities, which includes, of course, gene therapy, other approaches as well. But this is one that's gene targeted and non-invasive. And Matt Disney I see he's joined us. I know he joined a little bit. There he is. And other people at the Seaver Center have joined. Tess Levy is our clinical geneticist. Nan, I've introduced. Joyce is right on this slide here. She's the coordinator for the RDCRN study. And Paula is also kind of helping. She's a senior person in the wet lab side who's helping orchestrate some of these studies. Jess, you have questions?
Yeah, so I just wanted to recap for anyone who got lost during the presentation. So the VEP study is open now and it's recruiting now. So anyone who wants to and can travel to the Seaver Center can reach out to Stella and enroll. Is there any quorum of people with SYNGAP1 that you think you would need at a minimum in order to have meaningful outcomes from that study?
Yeah, so for this pilot study, we're really looking for 5 to 10 individuals who are local, who we can get a readout on. Like I mentioned, sort of these VP signatures that we're seeing in other disorders are very clear, that it allows us to start to sort of get excited about potential differences even in smaller samples. And, based on that, then as families come in for RDCRN, we'll continue collecting this data. But in terms of sort of the immediate need or desire, it would be to get about 5 to 10 local families in.
Okay, so we should aim for 10 local families, and the
great.
age is 18 and under, or is it under 18?
Anyone under 18, just
Under
for
18
purposes. I can send you the flyer also.
Okay. And we do have, I think one of our patients is a small baby. I think the parent might be on the call. But you said that it even is used in infants as well.
It is, I think, depending on the age. Yes, we have consent to do it at all ages, but we can talk with them and see whenever they'd like to come in.
Great. All right, we will work on that. And then for RDCRN, when we email Joyce, I think she's on the call, that's simply to register interest in being contacted in the future to participate in RDCRN.
Exactly. So what we would do now ahead of time, we'll collect the genetic reports, right? We'll do our phone screen. So that way you're already in the system. And then as soon as we're ready to enroll, we'll reach out for scheduling.
Okay.
But they're not, obviously somebody who comes to the sensory can also be in the RDCRN. And just to make it clear, we've been doing the RDCRN, as you've heard, for already 10 years. So all the protocols and all the personnel are in place. Simply a new grant, the paperwork has to be reactivated for the new grant. So we don't expect any problem whatsoever. It's a central IRB that we've signed off on and so on. So the timeline is quite solid. And I say that knowing that often it isn't so solid.
And both of those sites are at the Seaver Center, which is on the Upper East Side of Manhattan, which is just off the park, right?
Yeah, exactly.
Both at the same site. Okay, great.
And it's all the protocols go through a single door. So it's all
Okay.
single.
So once you've registered for one, the researchers can include you in the others quite easily. Okay, perfect.
Exactly. Yes.
Thank you.
If you consent.
Yeah.
If you can.
Yeah. And for example, the SAND is a measure in both studies. You wouldn't do it twice. It would just double count for each. So yeah, we work together on all of these projects.
Is that true? I'm sorry, this is Mike. Is that true just of the SAND, or is that true of other things? Because we have horror stories of patients going and consenting to multiple studies and being actually asked with a straight face to do the Vineland twice.
That will never happen at our site. As Joe mentioned, I think something very unique about our center, it is a single door of entry.
Yeah.
With only, one, our group of clinicians is the same. I oversee the study, so I make sure no one gets anything twice, but there would never be a scenario where someone would have to do something twice, unless it was because too much time had elapsed between the last one, for example.
Great, good answer.
And
I just,
likewise, sorry, with the physical tests, so if someone is coming in for RDCRN, can they do VEP at the same time? And just the reason I
Exactly.
ask is
Yeah.
with the
Oh,
kids,
yeah.
it's quite
So,
difficult to get them into the clinic
yeah,
sometimes.
yeah, yeah. No, we combine our study visits. So yeah, we wouldn't bring you in. Obviously, we want to get people in sort of now who are local while we're waiting for our DCRN to start up. But once it starts up, it wouldn't be separate visits. It would just all be included, and parents could choose to consent to both, and we would do it all within the same visit period.
Yeah, but the RDCRN is rare, almost never a single visit, right? Because
Yeah.
it's over the course of a couple of days for everybody's benefit, including the individual with the syndrome, right? And so one can easily do the VPs now and still do the rest of it. It has to be somebody who's local or able to.
yeah, I just want to ad lib for a minute, if that's okay. For the New York families who are watching this now and who will watch this later, I think this is a big opportunity. I think what you saw with Phelan-McDermott syndrome, which is also sometimes called Shank3, you might have heard us talking about Cure Shank3, and then ADMP, like what you've seen here is the team at Mount Sinai has done a lot of deep work. And for our SINs, it sounds like Syngap has hit their radar. And so that's a win. That's a win. You have Joe Bucksbaum, who's senior and well respected, and you have this team of other people, Tess Levy, Dr. Yang, Dr. Seiper, who are now interested in Syngap. This matters. And I'm focusing on you, Dr. Seiper. Am I saying it right? Is it Seiper?
Yeah, yeah, yeah.
Because you're a psychologist, right?
Yeah.
And as Syngapians get older, the psych issues get more severe. And, I was on a call this morning with Amber, and she has this great Venn diagram that I'm going to steal, which was like neurology, psychology. And where those two intersect, she writes SLC 6A1 hell. And I'm just going to replace that with SYNGAP1 hell. And I think, yes, we should do all these studies and we should advance SYNGAP science and more molecules are good. It's inherently good, but I just want to remind us that developing a community relationship with a team this robust it could be a long-term win for the community in terms of the more patients Paige sees rip electrodes off their head and try to trash the Seaver Center, the stronger opinion she's going to have about what meds work, what meds don't, and how to support our families. So I just and then there's Allison just went on camera. There's also the very serious issue of I'm losing my words, but intractable seizures, right? So I just want to encourage all the New York families to realize this is a opportunity that has been years in the making. We began investing in getting the DSC3 application years ago with your donations. Nurin, paying attention, the biotech interest is good. All the other stuff is good. When doors open, it's on us to run through them, and if we don't, other people will. So just a big thank you to the team at Mount Sinai and a strong encouragement to the New York families to take advantage of this opportunity.
Yeah, and I just wanted at the end to leave some time for the New York families to ask questions, anything that's top of mind, or even if it's just to give a bit of background about yourself and your child. Definitely love for you to come on camera and introduce yourself. And when I say New York, I mean metro area, people who can travel to the Seaver Center.
Hi, I'm happy to go first. My name is Allison. I actually live on the Upper East Side, so very close. My children were born at Mount Sinai East. I have three kids. My oldest is 8, and I have 5-year-old twins, Emma and Dylan. And Emma is my Syngapian. She's turning 5 in the summer. As Mike mentioned, when I came on camera, my daughter has intractable epilepsy. She experiences approximately 100 drop seizures a day, if not more. And we've tried and exhausted every single medication possible. And so we are definitely very much waiting for some miracle drug to come out to help her. She's nonverbal. She uses an AAC device. She actually I mean, I'm sure you guys are familiar with the schools in New York. She just got accepted to Gillen Brewer, which she'll start attending there in September. But it was a long and very, difficult process because Emma does need a one-to-one because of her seizures that are so dangerous. She gets injured very frequently. I don't know what other information I can share, but, we're very close. So I'm very interested in, joining and participating and doing our share, whatever we can do to help.
Thank you so much.
Yeah.
Thank you for sharing.
I can also go. I can introduce my daughter. My daughter's Hannah. She has mosaic SYNGAP1. We used to live on the Upper East Side. We were forced to move because she had to go to a specialist school, and the one that she was supported to go to, Tiegerman School, is out in Nassau County. So we had to move to the border of Queens, literally opened the map, wrote down where we're going to live, and went to see rental places out there so that she could have a reasonable commute out to Nassau. As you know, there's not many schools left in New York that are funded that can support our children, and most of the parents have to sue for FAPE, which I think Allison's probably going through at the moment with Gillen Brewer. So we would have definitely been able to come much more easily, but we can still travel in. My daughter is verbal. She doesn't have seizures. She's one of about 5 mosaic children that we know of with SYNGAP1. None of them seem to have developed seizures even at older ages. So something that we're crossing our fingers for. We still have to go every year or two to NYU to get an EEG. They let us take that home for a couple of days, and then we return the device after a few days of monitoring her wearing it. She has speech. It's probably similar to Mike's son, Tony, that she can talk, but not about things that have happened in the past very easily, loses context very frequently. She has a lot of sensory issues. She has severe behavioral problems, focus, attention, all the good stuff that comes with these syndromes. She's dealing with, even with apparently increased level of SYNGAP. So it kind of just goes to show you, even when you increase the amount of SYNGAP, any loss of it is pretty significant for the children. But we're, again, very excited to join. We'll be in on the Long Island Railroad and the subway. And
thank you. And we're happy to coordinate with days off from school and all those things to make it.
Yes, exactly. Thank you.
Thank you. You see Emily's hand up.
Yeah, I'm not sure if can you guys hear me?
Yeah, we can hear you.
Yeah.
Okay, and I think my video might be off, but that's okay. So I am from Boston, and my daughter Hadley is 8, and she is in the early exon. Let me see.
Rachel, I think you went.
Okay, can you see me now?
Yeah, Emily, I can see you now. Yeah.
Okay. So yes, we live here in the Boston area. My daughter is 8. Her name is Hadley, and she is on the early exon. She's on the 4th exon with the. We've identified this batch of kids in the early exons as milder. So Hadley is verbal and toilet trained and seemingly sometimes closer to a 5-year-old, but behaviorally really, really, really tricky, and does have epilepsy. She has good control with one medication, but had a bad breakthrough seizure when she got COVID from a fever. Actually, it was a low fever, but COVID triggered a tonic-clonic terrible seizure recently. So just curious if that would, the early exon children should be adding to the panel of kids if you only want about 10, or are you just looking for a minimum of 10? Do you want everyone of who can make it?
Yeah, I mean, we're interested in also just differences, right, between sort of the underlying genetics and what those VPs look like. In Phelan-McDermid syndrome, it's been really helpful because we saw sort of the correlation with the larger deletions and the larger abnormalities compared to the point mutations, for example. So we're open to all comers. I would say we have a collaborator in Boston that has the same system that we're using as part of another collaboration with Neuron. So we could definitely talk before you make the trip to New York.
Okay, because Dr. Wiltrout is our doctor at Boston Children's Hospital. She mentioned something was coming soon. I don't know if she's part of that.
Okay. I was gonna say there's a bunch of things happening, so we'll definitely connect offline. And the plan is if there is a closer site that we'll be able to send you to that. So for these initial ones, but I appreciate it. Definitely connect with us, but we'll try to bring in the families where travel is easier.
Okay, great. Yes, travel is our biggest challenge. We just got back from North Carolina and the airport was an absolute nightmare. So less is more. So thank you. Thank you very much.
But we do think genotype phenotype, which is the fancy way of saying what we just said, is very important, and we always try and take all comers. Obviously, you made a good point. We don't want to take so many, so diverse that we don't learn anything, but we want to capture the dimensions and understand. Even if you think about EEG or these VEPs as a biomarker, you need to know that they apply across the mutation spectrum. Carrie.
Hey, good afternoon. Thank you. I'm Kari. I'm moving from southern New Jersey to central New Jersey, about 20 minutes out of Newark currently right now. So I'm also pretty close here in New Jersey. Trajan is 10. He was diagnosed about 2 years ago with SYNGAP1. He's nonverbal, he's autistic, he has a lot of gait and ambulatory issues, and has a very, very interesting relationship with pain. He doesn't feel pain, and then he does, and it is a big deal for him. He's currently in a special school here in New Jersey that suits his needs very well, and I'm keeping my fingers crossed that I keep that school as I make this house transition. And he receives OT, PT, speech in school, and we receive speech both from CHOP, learning his AAC, and to sign, as well as speech outside of school, just to keep up with my sign. Because that seems, although he's really great with his AAC, he seems to really enjoy sign. And he only has one word right now, it's mama, but we work on a lot of approximations right now.
Best word to have.
And he started his seizures at age 3. And they're fairly maintained with his combo of medication right now, but he's recently had a couple of breakthrough seizures this year, which we're unfortunately dealing with and tracking really closely. He also has EOE, which keeps us on our toes, and he has a feeding tube to combat his EOE. And I'm looking forward to supporting both studies if at all possible.
Thank you. I would love to have you all. And also just so impressive, all of the services and schools you guys have been able to manage to find great support, it sounds like.
Yeah, we're working with our ABA. ABA falls in and out, but very grateful for a current ABA service right now too.
That's great. Nancy.
Hi, I don't know if you can see me.
Can't see you, but we can hear you well.
Okay, great. Okay, so let's see. My sister Karen just turned 70, and she was diagnosed with SYNGAP1 when she was 64, so just about 6 years ago. She is verbal. She's been verbal since she was little. She's had just such a lifetime of so many twists, turns, changes, and things. Right now, she's currently in a residential program. It's a day program with nursing and medical. It's a medical day program where she gets OT, PT, speech, and doctors and nurses. And the same facility that she's in has a group home that she's in, and she has 24-hour nursing there in that. She used to be able to walk, but she had a lot of gait issues and ambulatory issues. She's in a wheelchair now. And as far as sensory goes, sensory's always been an issue for her, especially she doesn't really feel pain, but if you touch her arm, she'll scream. So a lot of inconsistencies for her. And other sensory issues, like she can hear things. She hears everything going on anywhere. She can't seem to funnel those sounds out. But I don't think we're able to actually get her to come in to the facility. I would wonder if there's any way to do things remotely for any part of it. We'd be happy to participate. It's just really difficult because her physical needs are so big right now. We have to get several people to bring her. She's actually using a Hoyer lift at this point too.
We would love for you to just reach out to connect, remind us
Okay.
the connection. And, that way, as things come up, we do, have various remote studies, especially ones where we're actually looking into other syndromes. We recently published on InfoxP1. We're doing it in DDX3X, where we're looking at sort of the post-puberty into adulthood, psychiatric manifestations and development. So we would love to have you in our network, and we'll definitely call on you when we're at a place where we can get information remotely. It's definitely very helpful to get that full age spectrum.
Right.
And these
Thank
studies, we think of these studies as just the beginning. If we're going to commit to a gene, so we tend to do it for forever, right? And so new resources hopefully will come in, new studies, and so as Paige said, just kind of being part of our network, the opportunities will come up.
Okay, are there any more families who'd like to participate? I
Hi.
think Rachel came
Yeah.
up. Hi, Rachel.
I can't figure out my video, so I apologize. But yeah, so I'm in New Jersey. We're in Northwest New Jersey. Our daughter Grayson she'll be 10 in September. We go to CHOP currently, so we work with the neurologist there. She's in the school in our district. She gets all her therapies at school. They have a good program that she's in. So luckily we get all therapy there. We don't do any outside therapy except for equine therapy right now, which she loves. Her seizures are very much under control now. She is on the Ravicti, and with a couple other meds. So that's worked out really well. Behaviors come and go. Right now she's doing pretty well. But at any given moment, can go bad, as we all know. But she's getting older, so she's approaching the preteen. So a little worried about that, but we're willing to travel. Like I said, it takes us about 2 hours to get to CHOP. So I'm assuming I haven't Googled it to see how long it would be to get to Mount Sinai, but we're willing to travel if, as long as everything's in one day, we can make a day out of it. I did have a question though, and I may have missed it. Is it we go for initial and then it's like is it every 6 months or every year that we come back?
Yeah, good question. So the initial sensory study is just a one-time 1-hour event. So I think we'll definitely start with the people who are more local and then, combine it with the RDCRN visit. Our RDCRN visits, there's the sort of initial visit, and then 1 year later, there's the follow-up. The follow-up visits are 1 day. We're still waiting to see the completely final protocol for that initial visit. We historically have spaced them out over 2 days, which just tends to be easier on families for the most part. And as I mentioned, there's travel is paid for. Some families are staying in hotels for the night. But once we have the final battery, we always talk to the parents to sort of get to know them and their kids. So for some of them, the kids are able to separate for their testing, and a one-day visit doesn't seem overwhelming. For others, it's easier to sort of space it out. And so we will assess sort of the specific schedule on a day-to-day basis. We used to do these over 3 days, and now thanks to Zoom, we can do some of the parent interviews remotely still. The Vineland, I know, came up, for example. So we do try to minimize burden as much as possible, and use the time in the clinic for the things that we really need the kids there for.
Okay. Thank you. That's all I have. Thanks.
I just wanted to ask, because we're in New York City, what the level of support is for language and cultural requirements. If we have patients who would prefer to speak in Spanish or Chinese or Hebrew, how they're supported at the clinic.
Yeah. So we have, depending on sort of what the study is, I'll see what the RDCRN is, because it's a central IRB. We have consents in English and Spanish for most of our studies. We have translators that we can certainly use for many different things. The biggest thing for a study like RDCRN, where we have standardized assessments, is that the child speak or be exposed to English, because the norms on these assessments are generally English norms, or norms from when it was administered in English. But we do have a Spanish speaking coordinator at all times. So, we have the ability to sort of communicate with families who speak different languages. It's really just comes down to whether they have the fluency to consent.
And I speak both Hebrew and Chinese, but I don't think I'll be very helpful.
Okay.
But,
You're not in the official translator role currently. Okay.
But yeah, Sinai has a great translation system. So we, as long as we can consent and we don't have concerns about the child's, we want to make sure if we're measuring receptive language, that we're measuring it as accurately as possible, for example.
Okay, great. Are there any other families who'd like to ask any questions, anything top of mind? Certainly after the call, we'll distribute the email addresses that were flashed up earlier, so that you can get in touch, and we'll be actively recruiting to these studies. One thing I wanted to mention, which you may not have heard if you don't follow Mike's podcast diligently, is that if you are still going to CHOP, keep going to CHOP, keep enrolled in the PROMISE study, because that longitudinal data is very important. So it's not a case of goodbye CHOP, I'm going to go locally to Sinai. This is additional ask of you. Please keep going down to Philly, and stay enrolled in the PROMISE study as well. And Mike, you can add anything to that you might want to.
No, that's the message. I mean, Catherine and I have a working document where we're working on our talking points for the, if data is gold, longitudinal data is platinum. And if you've already got a couple of visits at CHOP, please keep going. We want that data. You're New Yorkers. You guys are tough. You can do more. It's fine. You're geographically selected. Just tough it out. It's what you do. And
I just want to mention, if you have assessments that were done recently or are being done at CHOP or vice versa, we have consents that you can sign. So, you can, your reports will have the information that typically goes, to parents, but, we can request and they can request sort of item level data or information from any of the assessments that we're giving, so that hopefully that reduces any burden of potentially doing things twice.
Dr. Cypher, I have a question about that. So will your DSC, so sorry, I have to read what I wrote in the thing, RD and CR, what is it, RD and?
Yeah, DSC, same thing.
Okay, are those clinical visits then, and that we can have the data from them? Or so is it a history study where we don't get the data?
No, so they're all prospective data collection. So kids will get a cognitive assessment, they're going to get the SAND, they're going to get let
me put Catherine's question a little differently. Right. Catherine and I interpret each other for people all the time. We have a citizen natural history study that Dr. Wiltrout wrote a beautiful paper on. So we are in the habit of hoovering up patients' medical records and learning from them. When patients come in on a research basis, those visits, notes, and scores are not available to the medical record.
Correct.
And the
That's
RDCRN will be that, right?
correct. And families will get a report that have the scores that they can then choose what they want to do with those reports in terms of sharing. But like within, research institutions, we can get consent, for example, to share the details of the cognitive testing scores with the psychologist there who can interpret them. So then they potentially can use some of that data.
But also all the data in the RDCRN is available as raw data to all the sites, and we'd like and also can be made available to others.
Yeah, I think, I don't know who on your team is going to be driving this. You keep talking, Dr. Cypress, so I'm going to look at you, but whoever needs to build that bridge with CHOP, please let us help facilitate that and plug you into the right person over there.
Sure. Yeah, I'm happy to speak to whoever to find out what that battery looks like and see where there might end up being overlap.
Great. And then Andrew, we had one more parent I think who wanted to talk, but Catherine, go ahead.
Yeah, before that, let me just make sure that we, is there a way for SYNGAP1 to know who has enrolled in the DSC or in any of your studies or not?
Only if parents report it to you. So everything's
right.
confidential. So we wouldn't, send a list to
right.
you.
So for those of you on the call, if you want SYNGAP1 to be aware so that we can potentially manage the data and manage the information better, you should CC us maybe on an email, or tell us later, but they can't, just so you hear it here, they can't just tell us. And you don't have to, it's up to you.
I think better offline because our coordinators will take anyone off. That's not, if we don't have consent to have them on these emails about it.
Oh, I see. Don't email us. Okay. Don't email us at the same time.
Offline, just without us on it. Yeah.
Andrew?
So just want to say hi to everyone. Not sure if you can see me, but my son Mateo is 7.5 months now. He was diagnosed with the SYNGAP through the Guardian study, so we knew pretty early on. We were just at CHOP last week. We did the assessment, so we've been involved there. And okay, I think we figured it out there. So far, we found out that he's at 5 to 15% level right now in terms of development, according to the PA at CHOP. So we're really just monitoring. We've noticed some eye strange twitches recently within the past couple of days. He's been on the on Keppra twice a day, but nothing completely out of the ordinary. So we're really just monitoring, and we're really happy to be connected here and just learn more about this and learn more about what's happening and just monitoring and taking all the necessary steps we need to do now so he has a good future.
Thank you so much. Nice to meet you.
Did you say 7.5 months or 7.5 years?
No,
7.5 months.
Wow. Okay. Amazing.
Who's doing the CHOP assessments?
Oh, that's a great question. So my wife went to the first one. I just made it to the last one, but I believe it's
offline. But if you have a list of the assessments,
I'll tell you.
Virginia, are you here? Can you take that one? Virginia manages CHOP, and then Marta's also here, so you got the.
I am. I'm happy to send you the protocol with all the assessments.
Okay.
Yeah.
And I can just give you all a sense of where the overlap is and isn't. So
perfect. Yeah, we can connect offline.
Thank you.
And then for the visual study, do you do that over time?
So right now we're funded for a single time point. In the RDCRN, we do it at all the visits.
Mm-hmm.
But
I was thinking that would be really interesting for such a young child for the visual one. Yeah. To do it longitudinally.
Okay, We don't have any more parents, or I just heard someone.
I think
Actually,
maybe-
can I chime in?
Sure.
Oh, hi. Yes, sorry, I don't know if you can see me because I'm dialing in through browser. So apologize if you cannot see me, but yeah, thank you very much for all the introduction and all the information you shared. My daughter Ellie, she is 3 years old, and she was diagnosed with SYNGAP1 when she was 1 year and 10 months old. And currently, I think so her mobile skill is kind of a little bit better, so she can run, but she cannot jump. And her walking gait has been improved really well since we started her PT. But on the speech side, she's still nonverbal. And on the behavioral side, a lot of challenges, I think. So she can be screaming, crying all the time, has all the tantrum if she doesn't get what she wants. And this morning, I noticed that she started to bite herself when she is upset. So which is really sad for me. And and she has seizures. We give her 2 medications. So it reduced her. So she has eyelid fluttering seizures. We give her 2 medications to try to reduce that. But for now, she still have 5 to 10 eyelid fluttering that I can see every day. And she has sleep issue, which I don't know if that's part of her diagnosis or it's a side effect of one of the medications that she's taking. So she's also taking the sleep medication to help her keep sleeping during the night. So a lot of challenges, and so we are really appreciating all the information and all the studies that Mount Sinai is doing right now, and we really appreciate this opportunity. And we are local. We live in Long Island, Nassau County. Yeah, we can easily travel to New York City for the study, and we will be glad to register for all the studies.
Thank you so much. We look forward to meeting her.
Thank you.
All right. Well, thank you, Sinai team. Thank you, Jess, for the leadership. And thanks you families for all the sharing. This is amazing. I mean, we are gonna Syngap is not the fastest moving. We're kind of like second place, maybe. But the speed from 0 to badass is remarkable, and that's because of what we're doing here. Like if we can get this New York families and Mount Sinai together, Mount Sinai's got the playbook from PMSF, and now we just go and do it again, better, faster, sharper.
There's a lot of evidence that being a rapid adopter is better than being first. Being second is actually often best.
This has been my strategy.
Mike, can I ask a question?
Yes, ma'am.
Ryan is my son. He's 10 years old, and he was diagnosed at about 3 with SYNGAP1. I missed some of the study because I had to take another call before, but are they going to be like doing any EEGs or anything like that? Like we haven't been able to do anything like that for him for about 5 years now because he's totally petrified to get anything put on his head.
So we're doing them for those who can tolerate. I should mention, because I know there's a lot of seizure history. These are research EEGs, so they're not clinical EEGs. We're not looking at seizures, and there's no clinical results that get returned. But we get it on many individuals, but not all. So if this is something that would not be something that you think your kid would tolerate, that is totally makes sense. They still would be eligible for the RDCRN. And we attempt on everyone, and if they can't do it, then we want every kid to be comfortable, so we're not going to put them in a situation where they're not comfortable.
Okay, all right, thank you.
Not to disagree with that part, but it's also true the 3 electrodes are often much better tolerated than the nets. right? And I mean, it's one of the reasons we also stick to that as kind of a rapid and quick way to get data.
Okay.
Also,
And
the
just-
time.
Yeah, the time I was going to mention to get a sense of it. So we have one sort of more traditional 60-second run where it plays for 1 minute straight, but then the short duration run is there are 3 seconds each, and we have to get 10 of them, 10 3-second good trials, but they don't have to be consecutive. So for kids who can't sustain their attention, we need 3 seconds of attention at a time. So
And again, the reason that biotech is interested in that is because they have the same issue. If you can't actually get a measurement, how is it going to be a useful biomarker, right? So things that can go on quickly without not be too interventionist and get data very quickly are very valuable.
Yeah, I mean, I realize you guys have forgotten more about studies than I'll ever know, but when talking to the uninitiated, it might be useful to say incredibly brief 3-electrode EEG-like study. Because when you say EEG, we just imagine holding our children down as somebody plasters these damn things to their heads. And then, I mean, literally, I've been through the, I'll never forget once I took Tony in, I'm like, dude, just be cool. And he kind of got the memo. He sat there, did all this to him at Stanford, 48-hour EEG. We drove home, get to the front door. I walk in the front door. He stops. I turn around. He just looks me eyeball to eyeball. Rips the whole thing off. Clumps of hair and skin with it. Just throws it on the floor. Steps on it once for emphasis. Walks in the house. I mean, we all have like EEG trauma. So maybe just say
helpful feedback, especially in a group of families where you've had probably tons of clinical EEGs. This is a very different experience.
Yeah, especially if there was some nice pictures you had on the presentation of the 3 and the girl with, the back of her head. That's very useful for us to share with the families, especially families that, they're not fluent in English. They can just see, okay, pictorial, that's what's going to happen. And it's going to take 15 minutes. And it's something that we can do social stories for the kids at home and say, hey, we're going to sit in the chair. The next thing that's going to happen is any social stories you can give us beforehand, we can distribute that to the families, and it gives them a lot more comfort that you know what a social story is and exactly what's going to happen on the day. So thank you.
I'll follow up with those.
Yeah, again, I'm allowed to say this because I don't do anything in the clinical space. So I mean, the team is exceptionally well versed and focused on profound autism, all the issues we're talking about. And we realize that we can't, we want to do research on those who are most affected. We have to develop the tools and the skills and the training to be able to deal with all these things. And that's one of the reasons there's also a single door. So everybody in the assessment group sees everything, and it's not like you come into there's some study just doing neuropsych, or everybody's an expert on all of these disorders and the phenotypes and the challenges.
I just wanted to briefly say hi. Sorry that I missed the beginning of this meeting, but I'm Tessa. I'm a genetic counselor and researcher at Sinai, and thanks for sharing all that information about your children. I'm excited to hopefully meet all of them in person.
And I would just say to that, genetic counselors have been the unsung heroes of progress for Syngap. So I'm really excited that you exist, because you guys are the smart, not that other people here aren't smart, but you guys translate. And I think a lot gets lost when families talk to people who have a lot of letters after their name. And so I'm really glad to see a genetic counselor on the team.
Tess is getting more letters as we speak, though. So I don't know.
Hopefully she'll stay good at what she does.
It's the hope.
All right. Well, thank you all very much.
Okay, and for the families on the call, keep your eyes open in all the forums where you keep your eyes open about Syngap, and we'll distribute the email addresses that you need to contact to sign up for the studies that were presented today at Sinai. And as always, if you have any questions, you can reach out to me, Mike, Lauren, Virginie, Catherine, or anyone that you know who does any kind of volunteering at SYNGAP1 can get you posted in the right direction. So thank you all so much for taking the time today. This was recorded. It should be up online soon, so you can review and share with the rest of your family who may need to watch it. So thanks again, and thank you very much to Dr. Buxbaum and the whole team at Mount Sinai for taking the time today. We really appreciate it.
Thank you for having us.
Thank you, everybody.
Thank you.
Thank you so much.
Thank you.